Yeah, so thank you to the organizers, first of all, for putting this very nice and exciting meeting together and for inviting me to join. So I'll share with you today some work that we've been doing in immunology, but you'll see how it connects to the broader themes of what we've been discussing so far, both on the level of protein evolution and then at the very end. I mean, I'm not going to talk about this in this talk, but I'm happy to talk about it offline. I think a lot of the questions that we heard about in the microbiome, thinking about population dynamics and ecology, can also be asked very nicely for the immune repertoire. And there's actually also now quite a bit of data, both on longitudinal sense and also across different individuals on immune repertoire statistics, where many of the same questions that we heard about in sort of microbial ecology are also beginning to be addressable. So today what I'll share with you is sort of more the protein evolution aspect of what we're doing in immunology, and I'll try and give you some statistical insights into the immune receptor code, and I'll explain a little bit what I mean by that. But basically what we'll be exploring is how can we extract something generalizable from having multiple instances of measurements of receptor to ligand binding maps. That's the sort of fundamental biophysical question that we are addressing. But to get us started off, I thought it would be useful, because I'm the first one, I guess, that talks about this particular biological system, to give you a very brief bit of background about the adaptive immune system. So here is the adaptive immune system at work. In purple here is a killer T cell that has recognized this cancer cell as presenting some antigen that it's specific to and that maybe shouldn't be there. And then you can see that it releases these cytotoxic granules to kill off this cell. So a very powerful effector mechanism that allows our bodies to clear, in this case, transformed cells, but in the case of, say, viral infection, a virally infected cell. And so with that comes the question of how does this cell interact with its target cells, and how does it knows what's going on within the proteome of the cell that it needs to decide whether to kill or not to kill. And so that's the question of how do these cells become specific to a certain target. And this, yes?
Maybe silly question about this movie, but is that time seconds, minutes?
I think that's minutes. I think it's not that fast, yeah. I think it's definitely much sped up compared to what's really happening here. Good. So how does it create specificity, and how does it create receptors that can specifically target, for example, in this instance, a mutated version of a protein that would be there in a normal cell? So adaptive immunity has come up with this incredible two-step solution for defending against new and unknown targets. So if you're thinking about other receptor ligand interactions, right? If you're thinking, for example, about the olfactory system, we have a set of olfactory receptor neurons that is coded for in our genome, which is a fixed repertoire of different olfactory receptor neurons. And then they have a relatively broad specificity to different ligands in order for us to be able to sense smells in a sort of combinatorial manner. Here, we have a different solution. So here we have much more diverse receptors, but then we don't actually hard code those receptors in our genome. But instead, what we have is a machinery that can generate cells with diverse receptors by genetic recombination sort of on the fly. So when T cells are first made from the progenitor hematopoietic stem cells, there is a process of gene rearrangement that can create sort of astronomically large numbers of different combinations by sort of playing Lego with different gene segments, and on top of that, at the junction of the gene segments, randomly inserting and deleting nucleotides. And so that's how you create diversity. The next thing is, how do you then make enough of the cells that are useful? And that's the principle of clonal selection. So you clonally expand the cells with the required specificity when they turn out to be useful. So there's an ongoing sort of population dynamics of the T cells and B cells within our body that reflects our past history of exposures. And so you will create more copies of the cells that have the receptor that, for example, can clear a certain virally infected cell or one of those cancer cells that you've just seen. And so what's very important with this process of creating random diversity and then selecting upon this is that this allows the adaptive immune system to have memory. And so this is a quote from Antoine de Saint-Exupéry, which sort of says, only the unknown frightens men, but for anyone who confronts it, it is no longer the unknown. And that's, in his setting, wasn't on the importance of vaccination, but I think it sort of really shows that this ability of the system to remember is at the heart of probably the greatest triumph of science over disease in general, with this vaccination, which with a single shot, right, not for all diseases, and there's a lot of diseases where there's still active research for making better vaccines, but for some diseases, it's really incredible, right? You give a single shot of, say, an inactivated version of the pathogen, your immune system remembers, and you're then protected, ideally, for a very long time against infection or maybe a severe disease. And this really relies on this sort of clonal selection principle and the fact that after you expand those cells, you keep some of them around as what are called memory cells, so memory of that infection in order to be able to mount a quicker response upon reinfection. And so with that, then, we're going to come to the heart of what we're going to talk about today, which is really the question of what are the molecular rules of the specificity of T-cell receptors. So we've seen that this cell interacts with these target cells, and it does so via two sort of transmembrane proteins. On the T-cell side, there's a T-cell receptor, which is a heterodimeric molecule composed of an alpha and a beta chain that interacts on the side of the cells that it's interacting with peptides. So these are small fragments of proteins that get chopped up and loaded into a molecule called the major histocompatibility complex, which has this sort of hot dog-like structure that you can see here, where there is a binding groove where you can fit in different peptides. And so these MHC molecules, they're presented on antigen-presenting cells that interact with these T cells, and so they will present fragments of all the proteins that are within the cell. So for example, if a cell becomes virally infected, some of the MHC molecules on its surface are going to be loaded with fragments, so small peptides derived from a viral protein. And so that means that the T cell is able to see that within that cell, there's an active sort of viral replication going on. Now, in the case of these responses to cancer, if these cancerous cells have some mutations, they can give rise to what are called neoantigens, so mutated versions of normal proteins that can also give rise to peptides that can be presented on these MHC complexes, and that allows the immune system, so T cells, the ability to survey cancer. And so you might have heard about the sort of recent successes of cancer immunotherapy, which really rely on unleashing these effector mechanisms again, because the cancer basically manages to tamp down this immune response. And so unleashing the brakes on that system is really what's now has been that revolution in cancer immunotherapy. So here then the question is, well, how does the specificity between the T-cell receptor and the MHC arise? And this is really a quite complicated question because it's really a very hypervariable binding interface. So on the one hand, we already talked a little bit about the fact that on the T-cell receptor side, VD-TR recombination, this sort of genetic recombination mechanism, can create very diverse receptors in particular, and I think this makes sense. This diversity is focused on the loops here that make the binding interface with the peptide MHC complex. In particular, there are sort of 3 loops for each of these 2 chains that are called CDR1, CDR2, and CDR3. So these are complementarity determining regions. That's what the name CDR stands for. And the primary sequence, so the sequence of amino acids within those loops, is what we'll talk about in the rest. And so those are very highly variable from one cell to the next that's created through this process. But this diversity is necessary to interact with a very wide diversity of ligands, because you can think about these peptides. So there's sort of two classes of presentation, MHC class 1 and MHC class 2. But for MHC class 1, let's say there's 9 mers, so then you already have turned into the 9 options. There, the actual MHC molecules, that's the most polymorphic locus within the human genome. So there's lots of different ones of the molecules of that flavor. And then for class 2, the things are even longer, maybe 50 mers. So again, you come up with very large numbers of potentially different peptides that can be presented. And as you've all known, now from the pandemic, right, we've all been able to mount both a T cell and a B cell response to SARS-CoV-2, even though SARS-CoV-2 was never around through all of our evolutionary history. So this is really a system that has a diverse enough set of receptors so that it can recognize basically any, first order approximation, at least any sort of ligand that you can throw at it. And so you need, in order to cover this large number of different ligands that you might want to detect, maybe that's why you need also a very large number of different receptors if you want to recognize them specifically. And so really what we'll be interested in today is, so there's one big question in computational immunology is, well, can you just from sequence information predict receptor ligand binding, which is sort of, some people have called that the holy grail of computational immunology. So can you use maybe a combination of sequence-based machine learning and maybe structural modeling to predict what receptor binds with what ligand? Today, we're going to do something slightly more biophysically inspired and maybe slightly simpler, which is we're trying to learn, address a slightly simpler question, which is, if you make one change there to a receptor that we already know that binds, what's the likelihood that that receptor still will bind the same ligand? So it's really about co-specificity. So we are trying to learn the rules, the local rules in sequence space of how much flexibility, how much degeneracy is there if we make some small changes. Do we retain specificity or do we change that specificity? So that's what we'll talk about. Yes, and so then the question is, so what does the kind of data look like? Maybe that's good to give you a sense of. Yes. Yes.
Like in each human, how many show up?
On the order of 100 million, so 10 to the 8. So it's a much, much larger space. And then in every individual, you'd have a much smaller subset. It's also a very unequally sampled space. So 10 to the 60 is sort of the richness, if you want, in an ecology sense. But the effective diversity in terms of a Shannon sense is maybe only like 10 to the 15. So it has a very wide distribution of, it has a very long tail of very rarely made sequences. That's the idea. But yeah. Yes.
In that sense, it also degenerates, like it can identify a lot of.
Yeah, exactly. Yeah, so it's a many-to-many mapping.
Yeah.
Yeah, so here we're going to really talk about the degeneracy on the level of different T cell receptors binding the same ligand, but you can ask the reverse question, which is how degenerate is the repertoire of different peptides that a single TCR can bind to. So if the difference between sort of the degeneracy of the solutions to one particular ligand problem or the degeneracy of the solutions to one sort of particular receptor. And yeah, you can, I mean, in the end, you have a bipartite graph, but it's interesting to ask questions about sort of the one-mode projections down to either of the two spaces. And here we're going to focus on the one down to the receptor level. Yeah. Okay, so here's the kind of data that's now become available over the last maybe 10 years or so, which is quite simple data in a sense. So basically what you do is you go fishing with a probe, which is basically a multimerized sort of version of your peptidymer C complex of interest. And so you take a blood sample where you have some cells in there, and then you sort out the cells that have binding specificity to your particular probe, and then you sequence the receptors of those hypervariable regions of these cells that you obtain. And so this is a little bit like what the data looks like. It's a particular visualization where what's shown is the pairwise sequence similarity between all pairs of CDF3 alpha and CDF3 beta regions. So I was telling you about the fact that it's this heterodimeric molecule. Just to make it a little bit more precise of what we're talking about here. So if we take one of these clusters, you can sort of make a little sequence logo of the sort of sequence variability within that. And so you see that you have some sort of local sequence variability within one of these binding solutions, but then there's also these clusters. So here's another solution to the same problem of binding the same ligand, that locally there's some variability, but then there's also globally quite different. And so these things here are basically one of those the loops here, the loop that is most proximal to the peptide. And so this is the primary sequence basically of amino acids within the loops of the cells that we've sorted here to be specific to one particular target. And so the basic question is, what can we learn from thinking a little bit statistically from this sort of matrix? Well, one thing direct that we see from this way of plotting things, right? So this is pairwise similarity of the alpha part of the sequence, or one of the chains, and this is pairwise similarity of the beta chains. And it turns out that they actually co-cluster with each other. So that tells us that pairing really matters. So if you have a certain alpha chain that will only work in the context of a certain complementary beta chain, and both can vary a bit, but they need to sort of be complementary to each other. And this makes sense structurally, because if you look at structures, so you see there are free loops, the way that these two chains come together, they're actually in structural contact. So we can look at here sort of a circus plot of where the contacts are. And you see that there's lots of contacts across CDF3 alpha and CDF3 beta. And so that constraint is a quite strong one that we sort of see directly just from this way of visualizing the data. But now what we have, and this is maybe where the statistical sort of physics and biophysics approaches come in, is that we don't have one such map, but we have many such maps for many different ligands. And we're trying to say something statistical about the structure of these maps that goes beyond sort of learning. You could learn, if you wanted, some sort of POTS model or something for each one of those maps. And you could really try, or some sort of generative model of other sequences for every one of those receptors. But what is interesting, because obviously this is still a very small part of the overall universe of ligands we would ever care about, can we say anything generic about the structure, the statistical structure that we see in these maps that would allow us to gain some general insights about, for example, questions such as, well, if I make a single change, what proportion of single mutations will maintain specificity? What's the fraction of functional sequences some distance away from a sequence that I know to be functional in sequence space? And so to do that, well, you need to think about this in a maybe slightly more abstract manner. At least that's what we've convinced ourselves that we should do. To really ask this question of, are there any unifying principles that govern these sequence to function maps? We should first introduce, yes. Yeah. Yes. So basically, maybe on this slide, maybe here, so basically what's shown in this map is pairwise, it's a pairwise distance on a sequence level, just here in a very simple manner. It's just edit distance, so number of edits that take you from one sequence to the other sequence, number of substitutions, insertions, or deletions. And if you have a pairwise distance matrix, you know it's symmetric, right? And so you can use that to display two different pairwise distance matrices. So one below the diagonal of the alpha chain and above the diagonal of the beta chain. That's what's plotted. Does that help? It corresponds to a particular sequence. And so every row and column basically corresponds to a pair of cells. And every cell has both an alpha chain and a beta chain. So below the diagonal, we show the similarity of the alpha chain of those cells. And above the diagonal, the similarity of the beta chains from that same cell. So this is roughly 100 cells, all experimentally sorted for their specificity to one given epitope. Yeah, so the interesting thing, why are we looking at similarity? So the interesting thing here is, and we're going to see this in a second, that this amount of similarity is much, much higher than the amount of similarity you would get just from bilateral combination. So, which I guess intuitively makes sense, right? Some local changes are likely to maintain your functionality. So it turns out that these things are really very highly enriched for similar sequences, and the precise way in which they're enriched for similar sequences is something that we're quite interested in, which is what we're going to get to. Good. Yes. Yes. Yeah, so these are the amino acids in the alpha chain and in the beta chain that are part of this particular cluster here of cells. So this is basically, this is the loop, sort of 15 amino acids long, something like this. This is the primary sequence within the loop that makes the closest contact with the peptide.
So, base pairing.
Yeah.
You know that it's just a complementary base pair, complementary base and it's close. Right?
Yeah.
So what are the rules for being closed? Yeah.
I mean, so far here, it's a very simple thing. It's literally just basic sequence distance, but we're actually going to get to more refined notions of sequence similarity in the later half of my talk. So I'm gonna get back to you on what are those rules. Yeah.
Is this not
Yeah.
You see that box structure at the bottom.
Yes.
What does that mean?
Yeah.
Sequences can be very.
Which these ones here, the ones that don't share similarity with anything else? Yeah, so I'm not actually going to get to this, but I do think we know parts of why that happens, which is basically these recombination probabilities range over like 10 orders of magnitude. So if you just happen to be a sequence that's specific, but your sequence that's very rarely made, you're just not going to have a lot of neighbors that are part of that same repertoire. So I think that the fact that you have this big blocks and small blocks relates a lot to the properties of actually the recombination process, more so than differences in the breadth of the binding solutions. At least that's what we think. Yeah. Good. Great. When did I start? Just because I'm sure of all this question. 5 past or 10 past? Okay, 8 past. I'm going to go with 8 past. Perfect. Good. Great. So we have many such maps. We're trying to learn some general rules. So let me introduce you to some notation that's going to help with the sort of statistical framework that is about to follow afterwards, which is the idea of, well, we have probability distributions, right? We have a generative, so we're thinking about sort of generative models, if you want, in the way that we've heard about also earlier today. And so we can think about the probability distribution of all the sort of CDF-free regions, if you want, right, of all possible sequences that can be made by Reddy combination. And we actually have relatively sophisticated models of those that really sort of capture the basic biophysics of sort of the combinatorial rules of these enzymes coming in and combining different segments and then doing various insertions and deletions. So we have relatively sophisticated models of the recombination machinery, so which give us generative models for the sequence diversity that we get just by genetic recombination. But then we have this sort of selection funnel and outcomes the sequences that are specific to one particular peptide MHC. But importantly, obviously, these sequences need to be made in the first place. So the fact that it's not a library, it's not an experiment where you made a uniform library to start with, and then you're selecting on that, but you have this highly non-uniform distribution with some complex structure already from Reddy recombination, and then you have some quite sparse sampling of from this sort of posterior distribution, if you want, which is sort of reweighted by these selection factors. Yes?
Your model does P of sigma include dynamic selection, or is that stuff in P of sigma?
You can, yeah, that's a good question. Yeah, so you can also make selection models for how to go from Reddy recombination to post-thymic selection, and then you can learn selection factors for that. I think for this, what we're going to talk about here, the differences there are much smaller than the difference we're talking about here, so it doesn't really matter whether you talk about relative to sort of the generation probability or relative to the post-thymic selection probability. But in practice, what we do here is we take the post-thymic selection probability as seen by just sort of taking a blood sample. Yeah. It's highly non-uniform, and one of the reasons it's highly non-uniform is that you choose these different genetic templated segments, right? So that's going to create some very broad global structure where you have different peaks in the landscape that correspond to being close to some of these germline segments. And then around those peaks, because of at the junctions, you do these randoms, insertions, and deletions, there you get sort of a spread. But you do have some global, quite complex global structure that comes from the different genomically templated regions. Yeah. That's the idea, exactly. Yeah. Exactly. But so here, basically what we have is we have these two probability distributions, and basically what we want to learn something about is some properties of Q. But what we have access to is some sparse samples from here, and maybe, yeah, we have samples from here, or we have also actual models that give us the probabilities. But the question is, well, how can we quantify basically some properties of Q? For example, we want to know something about the correlation structure in Q, and we want to learn this directly just from looking at this available data. And so what it turns out, what you can do, which it turned out to be surprisingly useful, is basically before I showed you these matrices, but you can just compress these matrices, the information in these matrices, down to a histogram of pairwise sequence similarities. So this is a probability of coincidence at a distance delta. So here's the sequence distance. Now this is here the sum of the distance on both chains, the alpha and the beta chain. And you can make a histogram on how often, if you take a random pair of cells that you know are specific to the same target, how often do they differ by 1 amino acid, 2 amino acids, and so forth. You can also actually resolve how often they differ by 0 amino acids because of the degeneracy of the codon table, right? You get different nucleotide level recombination events that actually convergently code for the same protein. So you can use that to actually get a sense of how often do you see exactly the same protein, but that has arisen through independent sort of recombination events. And then the other thing you can do here, and this is basically gets back to your question, why do we bother talking about sequence similarity? Well, if you do the same for like your null distribution, so what you would get just from P of sigma, it would be much, much less condensed in sequence space. So in the null distribution, it's incredibly unlikely to get sequence convergence sort of down to the level here, maybe like 10 to the minus 12. And what we see in the data here for this epitope that I just shown you is something like 10 to the minus 3. So there's 8 orders of magnitude of selection. So very, very strong selection pressure on the standing variation that has led to these observed sequences. And so then the next thing is, well, so looking at 0 tells us the overall strength of selection that has acted on these sequences. The other thing that's interesting, right, is that this enrichment extends to higher sequence distances. So you're also more likely, if you take a pair of sequences, to have basically these clusters, right, to have quite similar sequences, again, much more likely than in the background. And what you can actually show is that this, if you take the ratio of the orange to the blue curve, so this probability of coincidence of the distribution Q times P at the distance delta to the probability of coincidence of the background distribution at delta, that actually gives you basically the covariance of the selection factors for sequences at a given distance. So these near coincidence ratios tell us how selection co-varies with sequence. Or put another way, for example, we think about selection as sort of binary. You either bind or you don't bind. I mean, obviously, there is some sigmoidal nonlinearity probably that you could take into account. But if you think about it that way, basically what we're learning here is something, if we take some number of steps away in sequence space, what proportion of the sequences that number of steps away is binding the same target. And we're learning that simply by making histograms and basically counting how often do we see pairs of sequences at a certain level of similarity. And so you can basically now do this sort of construction, these histograms, and take the ratios together, these correlation function of these selection factors for not just one of these maps, but across these different maps. And so this is what this looks like here. So every gray line here is the way that this ratio of the probability of coincidence at a certain level of distance in the specific set relative to this null distribution how this ratio falls off with sequence distance. Yes.
Background is
Yes.
why does it fall so drastically? Basically, it's saying that you don't make two sequences alike very often.
Exactly. I mean, it's basically the fact that it goes up is basically a combinatorial effect, right? But the fact that it's very low here is just because of the diversity of sequences that you made, right? It's very, this thing here actually, I'm gonna spoil a bit what's coming later, right? But this is actually at distance 0, this is just the Renyi diversity. So this just tells you the Renyi diversity, sort of the order 2, the Simpson-Renyi diversity of the sequences is very high. So it's very unlikely to make exactly the same thing twice. It's not impossible, right? If it actually was 10 to the 60 equally likely things, it would be much, much lower still. It's actually sort of at the 10 to the minus 12 instead of the 10 to the minus 60, but it's still very unlikely that you make the same thing twice. So it turns out that at this level, there has been quite a bit of work that actually these distributions are relatively universal in terms of the recombination probability across people. So there's some variability, but nothing of the sort that we see relative to those. Yeah. So the post-trimix selection probability obviously has some imprints of the HLA of the individual, and there's a whole other talk I could give about that. But yeah.
So this, these are all receptors that are in the blood.
Yes.
Presumably from clones that expanded out.
Yes.
Does the size of the expansion matter?
Well, here, yeah, so these ones actually, they have both probably expanded in the individual, and then you go in and you sample. So there's a two-step selection process that we're grouping together. Especially for these ones, these are very immunodominant epitopes for chronic, in this case, it's EBV, so chronic virus that people will have seen before. So the idea is that this selection pressure that we're measuring here is the combined effect of in vivo selection and the in vitro selection step. Both of them come together. Yes.
Like the orange distribution has a little bit of a bimodality.
Yeah.
Right, one is sort of low sequence
Yeah.
distance and one is high sequence.
Yes.
Presumably, I mean, the second hump, the fact that it coincides with the blue is maybe just a coincidence. Right?
No, if we're going to get to that, that's one interesting feature of these plots. So yeah, the first interesting feature is just how much higher it is here. The other interesting feature is that you do get something here that basically looks like the background. And so that's the next slide.
Yeah, but it didn't have to be the case. Like you could have had human selection where you kind of suppress.
Exactly. So the simplest model actually, so this is the theory part of this slide. So here this is just showing that this sort of curve, if you take this ratio, it looks, I mean, obviously there's lots of variability, right? But it does fall off with a sort of roughly typical length scale across various different epitopes. So there's some generalizability, which wasn't directly obvious just from looking at the maps. The other thing is, well, and here just to guide the eye, actually all of this is sort of quite simple actually. I mean, here's just a function. It's not even a fit, it's just sort of by eye. So it falls off roughly exponentially by ratios of factors of 10 with every sequence distance. And then it plateaus out to basically be about the same as the background. So this factor of 10 here tells us, actually, if we take this idea of the sort of binary binding, it tells us that for every change that we make to a receptor, there is a roughly 10% chance that we are going to maintain binding specificity. And the fact that then it's roughly exponential, you can explain in a very simple model, where this model here is basically a model of just a sequence motif. So you just have a sequence motif, sort of an independent site model if you want, where at every position there's a certain number of good amino acids and some bad amino acids. And then with every change that you make, if you make a change at a position, there's sort of an independent probability that's going to take you somewhere where now that amino acid is one of the bad ones. And so you add those all together, you get something that's exponential. But what you get if you just have sort of a motif-like model is it actually sort of these probabilities go down and they keep going down. So this is, I mean, here just plotted out the six-mer, so we simulated six-mers with motifs. But if you have one fixed motif, you just get stuff that exponentially decays, but it's very unlikely to get stuff that's very far away. So how do we explain the fact that these curves flatten out? Well, what we think is going on, and I guess that was visually maybe already obvious in the earlier plots, there's multiple clusters, so there's multiple motifs. So you can reproduce the fact that it exponentially goes down and plateaus in these motif models by not having a single motif, but having multiple motifs, each of which has sort of a well-defined sequence variability, but the motifs globally are basically uncorrelated with each other. So that seems to be what's capturing what you picked up on, which is the fact that actually, interestingly, right, you get a second hump, which is about the same as the background. So that's what we think explains basically that sort of pattern. And so then, well, what else can you do? Well, you can actually link this, and I've already hinted at this. So you can link this back to information theory. So, right, minus log of this probability of coincidence is just a Renyi entropy of order 2. And so what we've been doing by taking these ratios of probability of coincidences between sets of sequences conditioned, so here pi stands for your PMHG. So if you condition on PMHGs and then you take an average over those and you take the ratio to background, well, that's basically your entropy of the total distribution minus the entropy of these conditional distributions. That's sort of a Renyi entropy equivalent of a mutual information. So basically what we've been measuring in doing this is how much mutual information is there between knowing the sequence and knowing its PMHC target. And what's kind of cute with this is that normally in Shannon entropy you can't do this easily, but here we have sort of an extension of information to something where I actually consider near matches. So you can ask how much information do I get if I know that receptor X and receptor X prime are like not actually quite identical, but they're identical within X number of delta number of edits. And then you learn about how much information do I get by having sort of a near match. And that is sort of a generalization, if you want, of information theory to non-exact matches. Well, the other thing you can do is actually, you can now use the language of information theory to look at some higher order sort of relationships between these different quantities. And so you can basically, well, we have these different sets, and we can actually take these various averages just by coincidence counting. We can actually estimate those entropies. And so in particular, I want to point out that often for Shannon entropies, because of the problems in how you estimate them from quite finite data, you maybe couldn't actually do this in this sparsely sampled regime. But with these coincidence probabilities, the sort of operational reason to maybe prefer them is that they actually become, we'll be able to estimate them from this relatively limited data, basically because of the birthday paradox, right? So if you have want to make sure you have at least 2 people that share a birthday, right? You need 366 people by sort of the pigeonhole principle, but the probability of 1/2 you already obtained with only like 20-something individuals, because you have the number of pairs that you're comparing is square the number of elements. So that helps. That together with this very stringent selection pressure allows us to actually estimate the entropy of a full chain, or even of the full pair chain of the receptor. And so we've done that, and we can then calculate how much information do we get about specificity from just knowing the alpha chain or just knowing the beta chain. And it recapitulates what we know about the structural biology of the problem. So the beta chain carries more information than the alpha chain. Another thing that's very interesting is if you compare the sum of these two to the off diagonal, which is how much information do you get by knowing both receptors, both chains at the same time, that is higher than these two by, I think, something like 4 bits. So that is an interaction information where knowing one chain and knowing the other chain together actually gives you more information than individually. And that goes back to those pairing constraints that we've seen earlier, sort of in these maps. And the other thing you can do is we've seen that there's some variability across different epitopes. And so here now we have sort of a local mutual information. So per individual epitope, we can calculate the information provided just by the alpha chain or just by the beta chain on the interaction information. And there's some variability in all these different values across different, so every point here is these quantities calculated for just the receptors that bind one specific epitope. And there's some variability, but most of that variability is actually explained by a single underlying degree of freedom. And you basically, these dependencies that we see between these different variables are again predicted by this mixture of motif models. So if you have a mixture of motif models, right, then if you have more motifs, we will get more less information on the single chain, but we will get more interaction information, because basically knowing where you are in one of the chains then determines the motif on the other chain. So that's basically, again, thinking about the key sort of generic thing to emerge from all of this is that we should really think about these distributions of receptors as mixtures of different structural solutions to the same binding problem. So with that, then I want to come to the question of, well, why does that work? And maybe also the question of what are the sequence similarity metrics that we should be using in this. So one thing that you could do is, and that maybe also gets back to the question of how much do you see just from timing selection? Well, if you have your TCR coordinate and your peptide coordinate, and you just look at the average selection factor across a very sort of rugged landscape, presumably, you will see something. Some receptors are just going to generically be better at binding than others, but you might expect something relatively flat, right? That doesn't carry that much information about very particular binding solutions, because they're all going to be at different parts of the receptor space and at these different coordinates. And so this actually turns out to be sort of what you see. There is actually relatively modest selection relative to generation probabilities in just the overall repertoire, for example, that we see in the blood. However, what if we sort of now align things in some way by a similarity metric, where we say we know what one receptor that binds, and we now look how far away am I from a receptor that binds. If you take a neuroscience analogy, that's a little bit like taking a spike-triggered average. So you align everything on a spike, you align everything else to that. But you sort of then would think that that aligns the different peaks, and then maybe what we show here, right, that then the distribution, even when you average over all possible peptides, has sort of a non-trivial mean field statistics that tells you about the average shape of one of those sort of peaks in this landscape. So that's the basic intuition. You can then turn this, well, if this is the case, right, then the idea is, well, can we sort of go the other way around, and can we actually infer good similarity metrics by looking at sequence variability that we see in these repertoires? And that's what I want to tell you a little bit about in the remainder of my talk. How do we now turn this around? Instead of just looking at these very simple edit distances that clearly, as we've seen, correlate with functionality, can we learn more refined notions of sequence similarity by directly comparing the observed sequence variability within sets of sequences that have the same specificity with our background expectation, again, like from VD recombination. And so first off, there's two different ways you might want to go thinking about this. One thing is that, well, what we did so far is we did sequence alignment, but what you might want to do instead is you can take in your receptors and you can embed them into some representation space and then measure distances in that representation space. And so that's something that's become very popular in the field, for example, with protein language models, so the same sort of attention-based models that we've heard about from Michael yesterday. You can also apply them to just sort of generic protein sequences, and you learn embeddings of proteins that presumably have, by some sort of mass language learning modeling objective, learn something useful about features of these proteins. And so that's the general idea. So can you use those protein language models to maybe measure distance instead? So we can take in the sequence, we can mask it, and then we can train a model to give us embeddings, where those embeddings are good at predicting these masked tokens. So we basically learn them by sort of trying to minimize the cross-entropy loss. And then the idea is that by doing so, you might end up with representations that have learned some useful features of these proteins. And you can then do transfer learning, and you can use those representations to tell you about the functional similarity within the receptor. So that's something that people in the field have advocated and also tried to apply to the TCR context. And it all sounds plausible enough, but the problem is it actually doesn't really work, at least not without fine-tuning. So if you just take these embedding distances of, say, ESM2 or PORT-BERT, which are sort of general purpose protein language models, or TCR-BERT, which is sort of a protein language model specific for TCR sequences, and you compare them on a task where you can compare them side by side with sequence alignment. So here the task is nearest neighbor prediction. You have some set of reference TCRs that you know is specific and some that you're trying to query against them. And in order to compare them side by side, let's not do, let's sort of really focus on the few shot setting, because that's anyway, for most of the things, we don't have many different receptors that we could train on. So let's really just focus on, are the distances directly interpretable as functional similarity? And the way to do this is just to say, well, my nearest neighbor in that set, according to this metric, how good is it to distinguish true binders from non-binders? And it turns out that it actually does worse than TCR-DIS, which is sort of the state-of-the-art sequence alignment-based method. It actually does, even a sort of TCR-specific protein language model does only about as good as just a simple edit distance on the CDR3. So clearly, it seems like this transfer learning doesn't work. And if you think back to the cartoon we had 2 slides ago, maybe one of the reasons it doesn't work is that what it basically has learned, right, it has just seen a bunch of different proteins. So it basically has learned this marginal distribution. But in this marginal distribution, maybe there's information about structure because of protein families, right? That's one place where people have used these protein language models successfully. But this for protein function prediction, it seems like that's not the features that are relevant for that aren't necessarily captured just by masked language modeling. So what we thought instead, what we sort of actually might want to do, we might want to learn in a contrastive manner good similarity metrics where we try and group together in our representation space sequences that ideally have the same sort of specificity and push apart the sequences that have unlike specificity. So you can think about this if you go back to our selection factors. We make some ansatz where our selection factors between pairs of sequences depend in some way on a trainable distance metric, and then we're trying to optimize this trainable distance metrics or parameters of that. And you can think about this as pseudo likelihood maximization. So over all pairs of observed sequences, what's the likelihood of the observed positive pairs relative to sort of a partitioned function-like term over all the negative pairs? And so you can derive this sort of loss, this contrastive loss function directly from sort of a pseudo likelihood maximization approach. Yeah, and so you can try and do this. Well, the first thing that you can try and do, you can try and ask, well, is there a way to use this to make these protein language models better? And actually, interestingly, it turns out the answer is yes, and even it's yes in a self-supervised setting. So what I just showed you, how I tried to motivate this contrastive learning is with the supervised contrastive setting, where you actually do know that those pairs of receptors bind the same ligand, and that's what you use for training. But you can sort of do a variant of this where you, instead of having 2 pairs that you know bind the same ligand, you actually give it, you make pairs of the same input and you try and re-embed those to each other. That's a self-supervised approach. So this is quite popular in image processing, say, for introducing invariances on your model that might not be encoded. For example, it's sort of a data augmentation strategy. For example, you give it a rotated version of the image and you want it to be robust under the rotations. You want it to re-embed that to the same point. But you can also do that much more generally, even just because the network path has some dropout noise. It will not actually embed even the exact same input to exactly the same space. So you can still use that contrastive loss function. It's what people call Siamese networks, because you sort of pass the same thing through 2 Siamese twins of your same network, and then you get 2 slightly different embedding points, and you try and basically make an embedding that's robust to whatever noise you introduce during that process. And so that's what we did for these TCR sequences. So we did a mix of this contrastive learning using a mix of sort of just the dropout noise, and also we can do chain dropping. We just give a single chain instead of giving both chains. And so basically at the end, what it does, it's creating a slightly different, it has a slightly different loss function for what a good embedding space should look like. And in particular, it has this part here, which is sort of this sort of a uniformity part, where it tries to use the embedding space quite uniformly, and that we think is what helps the model actually disentangle VD-Cherian combination effects from sort of maybe more specific binding effects. And so if you do this, well, you can now you're not just training these contextualized amino acid embeddings, but you're training an overall representation of the whole T-cell receptor that you train on this contrastive loss function, and we can then use that for comparing sequence similarities. And so we call this Receptor, a simple contrastive embedding of the sort of primary sequence of the T-cell receptor. And it turns out that if you do this now, at the very least, I mean, it's still not very impressive, right? Because this is just sequence alignment, but at least we're doing as good as sequence alignment. And so the next question is, so far, this is still a purely pre-training, right? It has never seen any data with actual specificity. Can we do better by fine-tuning it on data that actually, where we know the specificities of interest? And so yes, the answer is you can fine-tune these to really get very good at discriminating between a fixed set of ligands. But the amount of available data is currently such that they fail, these models, when you fine-tune them, at least in our hands so far, fail to generalize to the setting where you then test them on very disparate peptides. But instead, what we realized you could do as well with this very same contrastive loss function, you can also apply them to, in this sort of supervised setting, you can apply them to actually optimizing alignment weights. So this is a much more constrained model. So the idea is that it's maybe learnable from less data. So we can try, instead of like what TCR-dist, for example, does, it uses substitution matrices that are inferred from protein sequence alignment from general proteins that aren't T-cell receptors and aren't part of a binding interface. So it's using like these Blossom block substitution matrices. And we thought maybe that's not the most relevant substitution matrix for such a binding interface. And maybe on top of that, maybe we can begin to learn which particular residues matter more than others. And so that's just what we did. And so basically with the same, applying the same contrastive loss function with two trainable parameters of a sort of sequence alignment-based approach, we can recover a substitution matrix that is pretty dissimilar, I would say, from what we have in these evolutionary substitution matrices. In particular, we can reason a little, you see that here's this block structure. These kind of residues can all nicely substitute each other, and these can all nicely substitute for each other. And basically what this is capturing, and you can convince yourself of this by looking at the correlation of these matrices with matrices that you can get by looking at the compatibility of amino acids in terms of various biophysical characteristics, that basically Blossom is really capturing a lot of hydrophobicity of the amino acids that you substitute. And I think in these evolutionary sequence alignments that makes a lot of sense, right? So in these evolutionary sequence alignments, if you have a residue that's in the surface, then maybe you want it to be hydrophilic. If you have a residue that's in the hydrophobic core of a protein, maybe it needs to stay hydrophobic. And so that's a really strong constraint on what you can substitute for each other. On the other hand, in these binding interfaces, it seems like this hydrophobicity is maybe not as important a determinant of the substitutability. But instead, we see that, which we sort of see here, so our optimized matrix for this T-cell binding interface is much less correlated with these hydrophobic properties. And the things that it's most correlated with are some of the steric properties of amino acids. So it actually seems that shape matters. So maybe we're going back to the old Alan Patterson ideas of shape space if you want. So really if you're in a binding interface, if you have a very small amino acid, and all of a sudden you put something very bulky that probably has sort of a bigger, maybe also entropic effect on the ability to maintain that binding interface, then maybe the sort of more biophysical characteristics such as hydrophobicity or charge. And moreover, I mean, I'm not showing this here, but you can also begin to learn what part of the receptors matter how much. So particular residues, you can sort of learn a residue by residue weighting with the same approach that correlates with the how close the residues are on average in soft crystal structures with the peptide MHC, but not perfectly so. So even residues that are never in contact in soft crystal structures still can disrupt binding specificity, which makes sense, right? Because there's presumably in these loops quite a bit of epistasis. And we also see these signs of epistasis in the sense that residues not in direct contact do matter through their sort of indirect effects on the structure. And so with that, then I want to just wrap up by telling you a little bit about one of the reasons we're excited about these kind of approaches for measuring in a better way similarity between any pair of receptors, regardless of what their ligand might be, is that we can really use this for the unsupervised discovery of groups of T cells that have a shared function in otherwise very hard to interpret data sets. So in particular, you can take, so we're doing this now in the context of tuberculosis vaccine design, where traditionally the way vaccines are designed is you take an epitope of interest and then you test it out in humans, whether it's immunogenic, and then maybe you next test out, meaning do you get a good immune response, and then you next test out whether it's something that might be good for protection. But that's obviously at every step of that journey, you can it's quite labor intensive and you can go down a lot of false tracks. So the idea is, what if we could reverse this? What if we could study what a natural immune response looks like to see what are the kind of things from a bug that gets targeted most prominently in a natural immune response? And then can we use natural cohorts to look at the presence or absence of these particular types of T cells that target particular parts of particular epitopes from the pathogen to tell us which are more or less protective, and then take those forward for testing. So it's really reversing this process where instead of starting from an epitope and then trying to see whether it's immunogenic and protective, you're starting from studying natural immune responses to see what's immunogenic. You're then in the next step, you're looking at natural heterogeneity across different people in terms of how protected they are, and you try and link that back to the presence and absence of those immune responses, and then you take that forward for a vaccine. And that's particularly a useful approach for maybe something like tuberculosis because it's a bacteria, not a virus. It has like 4,000 proteins, so there's lots more different options for what you could put into a vaccine. And so the sort of traditional approach to vaccine design is maybe less readily applicable. And so what we're doing is we're basically getting human in vivo challenge data. So you can use these sort of tuberculin skin tests, which normally you use for diagnostic purposes. You can use them as a way of enriching for antigen-specific T cells in vivo. You can then sample those T cells, and then among those T cells, we want to identify groups of T cells that we think have the specificity to a common target. And so the idea is we basically can group T cell receptors together in this sort of conglomerate of all the cells that will respond to all the different antigens that are in TB. We can group them together. We can associate them with the MHC by looking at in which individuals they show up. And then maybe ultimately we can then go from the receptor. We can express those receptors in cell lines, and we can do screening against peptides together with sort of also more computational techniques to prioritize peptides for testing. We can then go reverse back and try and discover the epitopes that they target. So that's what we're trying to do now, and some pilot data is just on bioRxiv, but it's like a bigger long-term project that we have now. And then the last thing before I'm gonna shut up is I just want to tell you a little bit about some of the other scales. So here we really focus. So adaptive immunity, if you take it across, is really a multi-scale problem. And here I talk to you mostly about this sort of macromolecular level of the interactions between receptors and ligands. But I think there's also probably some interesting discussions to be had, maybe later today, or maybe over the rest of the 2 weeks, about some of the other scales. In particular, there's interesting questions around the population dynamics that relate to sort of the ecology, ecological questions about competition and about environmental fluctuations and how they shape the statistics of immune repertoires. There's interesting sort of quorum sensing and signaling questions. And at this level, there's interesting evolutionary questions about the design of these receptor repertoires and sort of how the evolution of these germline segments. And then questions on sort of more the level of building blocks, which is on, say, the process of Reddy combination. So lots of things to discuss. But with that, I want to acknowledge the amazing set of students and collaborators that were involved in this work. So particularly Yuta Nagano, who has led the work on the protein language models, Martina Milighetti, who has led some of the structural work, Andrew Pio, who has led the work on the alignment, contrastive learning of sort of alignment weights, James Henderson, who led the work on the information theory, and then 2 collaborators in particular, Kurt Callan at Princeton, who we've been talking with over a long time with this idea of what can we learn from these matrices, and then Benny Chain at UCL, who is a very close collaborator since I moved to UCL. And thank you all for your attention.
point what's model
Yeah.
they care about. But I was wondering if you have if you imagine that what's models are here actually able to do what to do with contrast learning.
Yeah, I think basically the idea, and I'm not sure whether people, I mean, people must have worked on this a little bit, is you have one pod model per group of receptors that binds one particular target, but what you basically want to do is a hierarchical version of that, where you somehow use the fact that you have seen other instances of such problems, so you want to regularize when you infer a new pods model on one particular sequence alignment for one particular ligand, you want to somehow use the information that you get from other such problems. So you have a multiple instances of trying to learn the same pods model, and you want to somehow use the structure. I'm sure there's some way to do that in a sort of hierarchical pods model framework, but I'm not sure I've seen people do that yet. Yeah, but it's an interesting question.
That some of the sequence differences are tolerated, like the receptor can still maintain its ability to bind, right? Would you say that it's more related to the geometry of the molecule, or as in the higher order structures, or is it there's no way to separate the geometry part from the binding affinity aspect?
Yeah, you mean in what step? Is it because the structure changes a lot, or is it because the structure changes but the binding interface doesn't have the right energetics? We don't really know currently in a very precise way. I think the fact, for example, that the shape of the substituted amino acid is a bigger determinant tells you that probably quite a bit of this has to do more with changes to the structure. And then maybe only as a second order changes to, do you still maintain specificity? That's would be my guess, but I'm not sure we have a very exact answer. It's sort of an open question.